welcome to this Integrated DNA Technologies webinar on knockdown of long non-coding RNAs, exploring RNAi and antisense oligo methods. My name is Dr. Hans Packer, and I will be serving as the moderator for today's presentation. The presentation today will be given by Kim Lennox, and Kim is a research scientist here in the molecular and genetics group at IDT, and she's been here for 13 years. She's an expert at using synthetic oligonucleotides to suppress gene expression and is a co-author on 17 scientific papers describing various methods of gene knockdown. Kim studied genetics for her bachelor's degree at Iowa State University, and she went on to receive her master's in science degree in biomolecular archaeology from the University of Manchester in England. Kim's presentation today should last about 30 minutes, and following that presentation, Kim will answer any of your questions. As attendees, you've been muted, but we encourage you to ask questions or make comments at any time during or after the presentation, and you can do that by typing them into the questions box, which is located at the right-hand side of your screen. At the end of the presentation, I will direct your questions to Kim, and we will take as many of those as we can. And in case you want to leave early, you need to leave early for some reason today, or you want to revisit this webinar later, we are recording the presentation. We'll make a link to that recording available a few days after on our website, which is idtdna.com, and you'll find that under our Support Education tab. Also, we have a YouTube channel, which is www.youtube.com/idtdnabio, where we'll have this webinar posted, and we have a series of past webinars that are posted already. And we also have a SlideShare site, which is www.slideshare.net/idtdna. So if you want to go back and look at the slides specifically, you want to see the slide deck, maybe pull out some references or whatever, you can go to that site and get those. So without anything else, I'm just going to hand this over to Kim and let her get started. Kim?
Thank you for the introduction. Today's webinar is going to focus on optimal methods to knock down long non-coding RNAs. I'm going to compare two different commonly used RNA knockdown strategies, RNA interference and antisense, for their ability to knock down long non-coding RNAs and discuss whether or not there's an advantage of one technique over the other, depending on the subcellular localization of the long non-coding RNA. Okay, so what are the importance of long non-coding RNAs? Well, there have been tens of thousands discovered with a wide variety of functional and structural roles in the cell. The few that have been functionally characterized have been found to play important roles in gene regulation and differentiation. This figure here out of Cell Cycle gives insight into how long non-coding RNAs might actually be the driving force behind species complexity. And so they have coding and non-coding base pairs here graphed against the genome size as well as number of distinct cell types. And so over on this side of the image are the single cell organisms, and over here are the multicellular organisms. And as you can see, if you look at coding base pairs, which are indicated by the red squares, you get an exponential increase in the number throughout prokaryotes. And then once you get into the higher multicellular organism, this effect plateaus out. What this means is that you can have similar number of coding base pairs in simple organisms as you do in higher multicellular organisms. On the other hand, the non-coding RNA base pairs, which are indicated by the blue squares, continually to exponentially increase with biological complexity. And this allows an organism potentially to achieve a greater number of diversity from the same number of protein coding genes. And so the question is this the driving force behind biological complexity? Is it kind of what makes humans humans and different from other non-human primates? With long non-coding RNAs continually becoming more interesting, being able to successfully knock them down to determine their function is becoming more important. And so knocking down RNAs or modulating their function has been done for a number of years. Historically, the original RNA is a messenger RNA that has been knocked down with antisense was the first technique. And the location of messenger RNA is both in the cytoplasm and in the nucleus, transcribed in the nucleus, and then it's shuttled out to the cytoplasm to be translated into its protein. And so these are considered to be kind of dual localized. Splice alteration using, for example, splice shifting oligonucleotides is something that has been achieved more recently, and if this is something that you want to do, this functions only in the nucleus. MicroRNAs are small non-coding RNAs which are involved in post-transcriptional regulation of translation, and while they are found both in the cytoplasm and in the nucleus, they are more abundantly found in the cytoplasm. And the long non-coding RNAs are in a unique category in that they have different subcellular localization depending on their function. So you have some long non-coding RNAs which heavily populate the cytoplasm, some which are more equally distributed between the cytoplasm and the nucleus, and others which more heavily populate the nucleus. This figure over here is from Cell in 2013, and this is RNA-seq data showing that typically messenger RNAs more heavily populate the cytoplasm, where when you look at the long non-coding RNAs, they're a little bit more diverse, where you have some which heavily populate the cytoplasm, some which are more equally distributed, and others which are found exclusively in the nucleus. A second study that was done recently in Genome Biology in 2015 is looking at RNA fish data on 61 long non-coding RNAs. More specifically, they are using optimized probe sets, the Stellaris probe sets from BioSearch, and they found a similar story where some of the long non-coding RNAs heavily populate the nucleus, others are more equally distributed, and then some are found to heavily populate the cytoplasm. So the two different RNA knockdown techniques that we're going to talk about today are RNA interference and using antisense oligonucleotides. So just briefly, RNAi works by using a small double-stranded RNA called a small interfering RNA. This is incorporated into the RNA-induced silencing complex, or RISC. Within RISC, one of the strands, the passenger strand, is discarded, and the guide strand is complementary to the target RNA. It binds with the assistance of this multi-protein complex, inducing RNA cleavage. Antisense oligos, on the other hand, have a DNA component. They are single-stranded short oligonucleotides, which use thermodynamics to target their RNA, so there's no assistance of a protein complex here as there was with RNAi. And then it recruits RNase H1, which recognizes a DNA-RNA heteroduplex and cleaves the RNA. And so this is kind of what we're going to be looking at is there a difference between these techniques depending to be able to knock down a long non-coding RNA, because typically this is an overgeneralization, but RNAi is thought to work more effectively in the cytoplasm because that's where a lot of the components are located, while antisense oligos, because RNase H is more abundant in the nucleus, is thought to function more in the nucleus. Now, this is not necessarily an absolute truth because the RNAi components are also found in the nucleus and have been found to be able to knock down nuclear RNAs, while RNase H is also found in the cytoplasm. And that's not the objective of the study, is not to prove or dispute one method or the other. It's just to kind of give a generalization as if you wanted to achieve the highest success in knocking down your long non-coding RNA, is one of these methods more optimal over the other depending on the localization. So the study design, we looked at 8 different long non-coding RNAs, 2 which are found more exclusively in the nucleus, 3 which are heavily populate the cytoplasm, and then 3 which are dual localized to be found both in the nucleus and the cytoplasm. And we looked at both antisense oligos and siRNAs. For the antisense arm of the study, 12 sites were designed internally by IDT, and 2 different chemical modification patterns were used, the DNA phosphorothioate and the 2'-O-methyl-PS gapmers. Additionally, 6 LNA long RNA gapmers were designed and ordered by Exicon. For the siRNA arm of the study, there were 12 dicer substrate siRNAs, which were designed by our internal algorithm optimal for dicer substrate design. We tested 12 siRNAs at different sites, which were designed by the online GE Healthcare design tool. And additionally, 4 silencer select siRNAs, which are LNA modified, were ordered from Ambion through Life Technologies. These are the chemical compositions that were tested. This first antisense oligo is considered our first generation antisense oligo. It's fully DNA bases with phosphorothioate modification or linkages, which serve to increase nucleus stability of the antisense oligo. Our second generation antisense oligo incorporates 5 2' methyl bases on the flanking ends, retaining this DNA central core, which is the substrate for RNase H. Again, it's fully phosphorothioated. The LNA gapmers that are used in the study are generally 15 to 16 mers, and the LNA placement is proprietary to Exicon. And so this is just a schematic of what it might look like with the LNAs on the flanking ends retaining the DNA core in the center. But again, the LNA modification placement is proprietary. The siRNAs that were used are double-stranded short RNAs, which have the 2 base 3' overhangs on both the guide strand and the passenger strand. The dicer substrates are 27 mer, which indicate the length of the guide strand. They're asymmetric designs in that one end is blunt. The other end has the 3' overhang on the guide strand. And on the sense strand, there are also these DNA bases here, which serve to reduce unintended loading of the passenger strand into risk. The transfections were done in both HeLa cells and in the HTT116 cell line, and the data were seen to be concordant between the 2 cell lines. All of the images that you see today were performed in HeLa cells. With the exception of NRON, which is one of our cytoplasmic targets. These were performed in HUH7 cells because it was lowly expressed in HeLa cells. All of the transfections were done in biological triplicates in 9612 format using lipofectamine 2000. For the negative control sequences for all of the oligos that were synthesized by IDT, so the dicer substrates, the 21 mer siRNAs, and the longer antisense oligos, I used either 2 or 3 different negative control sequences, all of them which matched the chemical composition of the oligo being tested. The Exicon LNA gapmers were compared with the Exicon negative control, and the Ambion silencer select siRNAs were compared to the negative control purchased from Ambion. RNA was prepared 24 hours later, and the qPCR assays there were 2 tested per long non-coding RNA, one at the 5' end and one at the 3' end to verify concordance between the 2 assays. All qPCRs were performed in triplicate in 384 well plate format, and results were normalized against both HPRT and SFR9. The long non-coding RNAs that were studied varied in cellular localization as well as in length. The nuclear targets are MALAT1 and NEAT1. The cytoplasmic targets are NRON, DANCR, and OIP5 antisense 1, and the dual localized long non-coding RNAs were TUG1, Cas7, and hot air. The longest long non-coding RNA looked at is 9.3 kb in length, while the shortest one was just under a kb in length. There is a lot of literature which indicated localization of some of these more studied long non-coding RNAs, but we also wanted to internally validate their localization using in situ hybridization. The method that we used looked at one site one different in situ hybridization probe, which was dual labeled with DIG using Sci5 detection methods. And I'm going to show you a couple examples here. For MALAT1, you can see In most cases, it's exclusively found in the nucleus, especially in cells which aren't actively dividing. The same result was seen with NEAT1 in that the localization was mostly found in the nucleus. Cas-C7, there wasn't a whole lot of literature on the localization, and so we definitely wanted to verify this one internally, and we found using our in situ hybridization probes that Cas-C7 is dual localized in both the nucleus and the cytoplasm. For the knockdown studies, all of the graphs are going to be organized similarly, where the results are expressed as a % remaining RNA, so these are actually knockdown levels. Two different doses are going to be shown, 10 nanomolar and 1 nanomolar. On the left side of the graph are going to be the RNAi reagents, starting with the dicer substrate RNAs. Next is the 21-mer siRNAs. In the middle are the Ambion silencer select LNA modified siRNAs. On the right-hand side of the graph are the antisense oligos, starting with the 2'-O-methyl PS gapmers and the LNA PS gapmers ordered from Exicon. On the x-axis, we have for each class of reagent, we have them organized from 5' end of the long non-coding RNA to 3' end of the long non-coding RNA. MALAT1 is our first nuclear target, and as you can see, while we did find some reagents to knock down MALAT1 for the RNAi reagents, overall, the antisense oligos outperformed the RNAi reagents for knockdown, especially at the 10 nanomolar dose, where almost all of the sites tested gave about an 80% knockdown or greater at the 10 nanomolar dose. Our second nuclear target, NEAT1, gave more striking results in that it was even more difficult to find an RNAi reagent to knock down NEAT1. There was a couple which knocked it down maybe 40%, but overall the antisense oligos outperformed the RNAi reagents at the 10 nanomolar dose, with showing most of the sites having 80% or greater knockdown levels. NRON is our first cytoplasmic target that was tested. Again, this was performed in the HUH7 cell line. And interestingly, we didn't find any of the knockdown reagents to be able to degrade NRON. This was repeated 3 times, and our positive controls in all the plates that we ran were fine. And so we believe this result. And when looking more at the function of NRON, it serves kind of as a protein sponge, where there's 11 putative proteins which bind to NRON. And if an RNA heavily binds proteins, this could make it inaccessible to an antisense oligo or for the guide strand of an siRNA to be able to bind to NRON. Again, this is all speculation. Otherwise, NRON could be localized in a part of the cell which has low abundance of RNAi factors or RNase H. But regardless, we found it very difficult to knock down this long non-coding RNA. So we moved on to a second cytoplasmic target, DANCR, which gave a completely different story. This one was very effectively knocked down by RNAi reagents, and it showed greater performance overall than the antisense oligos. However, the antisense oligos were active at the 10 nanomolar dose in several different sites. What is not seen on this graph is when we lowered the dose for the RNAi reagents, we had to go down into almost sub-picomolar doses before we started to lose activity. And so that was really the determining factor in showing that the RNAi was significantly outperformed antisense for this particular cytoplasmic long non-coding RNA. We saw a similar pattern for the second cytoplasmic target, OIP5 antisense 1, in that the RNAi reagents outperformed the antisense oligos. However, again, we did find a lot of active sites for antisense oligos at the higher 10 nanomolar dose. But at the lower doses, the RNAi reagents outperformed the antisense oligos. For our first target that is found both in the nucleus and the cytoplasm, we found that both techniques worked. The RNAi reagents were only able to knock down TUG1, however, by about 60%. And you see this plateauing effect instead of a nice dose response. And this was also seen at lower doses, which we found to be interesting. The antisense oligos, on the other hand, continued to have a nice dose response, again finding some very active sites at the 10 nanomolar dose. And so this kind of asks us the question of whether or not there was a pool of the TUG1 RNA which was inaccessible to the RNAi factors. We saw a similar effect with Cas-D7 in that, for the most part, we were able to knock it down maximally at 60% with the RNAi reagents. The silencer select performed a little bit better here. We again saw the plateauing effect even at the lower doses where once maximal levels were achieved, it was harder to get further knockdown, where for the antisense oligos, we saw the nice dose response and found a lot of active sites at the 10 nanomolar dose. The last dual localized long non-coding RNA we looked at was hot air. Again, it gave a very similar story. This one was a little bit easier to knock down with the RNAi reagents. Most of them gave probably about 60% knockdown, some up into the 80% knockdown. Again, we saw the plateauing effect where for the antisense oligos, they didn't work as well at the 1 nanomolar dose, but at the 10 nanomolar dose, we found several active sites. So here's kind of an overall comparison of both methods using only the 10 nanomolar dose. At the top of the graph are the antisense oligos, and the bottom are the RNAi reagents. The antisense oligos, the green bars indicate the 2'-O-methyl PS gapmers. The blue are the LNA PS gapmers from Exicon. For the RNAi reagents, the red bars are the dicer substrates. The orange are the 21-mer siRNAs, and the black bars are the silencer select LNA-modified siRNAs. On the left-hand side, we have the nuclear targets. In the middle, we have the cytoplasmic targets, and on the right, the dual localized long non-coding RNAs. For each different long non-coding RNA, we have the knockdown reagent organized from 5' to 3' end of the long non-coding RNA. So you can see over here on the left-hand side, the nuclear targets, the antisense oligos, outperformed the RNAi reagents at the 10 nanomolar dose. For the cytoplasmic long non-coding RNAs, while the antisense oligos worked, the RNAi reagents significantly outperformed them, and this is seen again at the lower doses, which were not shown today. For the dual localized targets, we found that both methods worked, but the antisense oligos probably outperformed the RNAi reagents overall. This could be because the antisense oligos can also knock out or cleave an RNA before it's exported out of the cell into the cytoplasm. But after seeing this result, where there seemed to be a maximal amount of knockdown activity by RNAi, we wondered if combining the two technologies could get even enhanced knockdown activity. And so we selected a nuclear long non-coding RNA, a cytoplasmic long non-coding RNA, and one of our dual localized long non-coding RNAs. And we looked at an antisense oligo, a dicer substrate, looking at from 1 nanomolar dose to 10 nanomolar dose. And when combining the two methods for the nuclear target, we didn't see any improved activity for the higher doses, but then potentially for the lowest dose employed, there was a little bit of enhanced activity when combining the two methods. For the cytoplasmic target, when we combine an antisense oligo and a dicer substrate, we got enhanced activity. And for our dual localized target, again, an antisense oligo combined with a dicer substrate, we got improved knockdown efficiency after combination. The last thing that I want to discuss is a comparison of the chemical modifications of the antisense oligos. If you remember, I said that we tested DNA phosphorothioate, which are our first generation antisense oligos, and we wanted to see whether or not the 2'-O-methyl gapmers continually outperform the DNA phosphorothioate, and we also wanted to see how well, in comparison, the LNA gapmers functioned. And so we selected, this is for MALAT1, and we selected the 6 sites that were optimally designed for the short LNA gapmers. So these were designed by Exicon, and we converted them to 20 mers at the same site to the 2'-O-methyl gapmers as well as the DNA phosphorothioates. And we found for every site the 2'-O-methyl gapmers outperformed its DNA phosphorothioate at the same site, which suggests that indeed the 2'-O-methyl antisense oligos are more potent. For the comparison with the LNA gapmers, 2 of the sites, the LNAs, significantly outperformed the 2'-O-methyl gapmer antisense oligos. For the other sites, they seem to be equipotent. So for 4 out of the 6, they were equipotent, and then for the other 2, they outperformed them. One of the things that I want to point out is the disadvantage that we find with the LNAs is that sometimes they can cause more toxicity. Not all the time, but sometimes we've seen that, and it seems to be sequence dependent. One of the advantages of the LNA short gapmer antisense oligos is that they are more effectively taken up in vivo when delivered naked. And so if the intentions of the experiment are for in vivo naked delivery of the oligonucleotide, I recommend using the LNA short gapmers from Exicon. If you're going in vivo with the delivery tool, such as a lipid nanoparticle, the 2'-O-methyl gapmers will work with the delivery tool. So overall, we found that there was a difference between the efficacy of antisense oligos versus RNAi reagents, depending on the localization of the long non-coding RNA. The antisense oligos were often more effective at knocking down nuclear long non-coding RNAs, while the siRNAs were often more effective at knocking down cytoplasmic long non-coding RNAs, especially if a lower dose was employed. We also saw that you can achieve better knockdown if you combine the 2 techniques. But overall, if your long non-coding RNA is dual localized, the antisense might have worked a little bit better at the high doses. But this kind of shows us how important it is to characterize the localization in order to have a successful knockdown experiment. And if you're unable to characterize the localization of your long non-coding RNA, you may need to try both methods to see which one works better for your target. So additional articles about long non-coding RNAs can be found on our website. We have a couple that are specific to long non-coding RNAs, and then one that's specific to antisense oligo design. And if you're interested in designing dicer substrates, we have an online design tools available. If you're interested in designing antisense oligos, you can contact our application support at this email address, and we can assist you with your antisense oligo design. Thank you for your attention, and now I'm going to pass you over to Hans in case there's any questions that we'd like to answer.
Okay, thank you everybody for your participation, and Kim, thanks for the great presentation. We do have some questions, but before I get on to that, I just want to remind everybody that if you do have a question and you haven't already asked it, you can type that into the questions box in the GoToWebinar control panel, and there's a little plus sign or up arrow depending on whether you're on a Mac or a PC. If you click on that, you can make that window larger, type your question in. We've got quite a bit of time here to answer some questions, and I do have a list of them. So yeah, I guess we'll just get started with that. So one of the things, there was a good question that I know is relevant about structure, which is based on the observation that you had on slide 16, would you say that the potency depends on the secondary structure of the target?
on slide 16 here. Let's go back to slide 16. Yes, structure can very much impact the efficiency of a knockdown reagent. So when using RNA interference, there seems to be some assistance with the protein with risk in being able to unwind the target in order to increase the accessibility of that. While antisense oligos it kind of relies exclusively on thermodynamics in order to do so. When these were all designed, we actually looked at target structure using unifold and tried to design the antisense oligos in accessible regions that were already open to kind of try to limit that bias. But if you went in there blindly designing antisense oligos, yes, structure can definitely impede their ability to bind to and knock down the RNA.
And that was the second part of the question that the person asked, which was how could you predict them? So unifold is what you would tell people to look at?
It's difficult to predict easily computationally. And so I've had success using unifold, and so that's one of that's the software that I use, looking for accessible regions. And then after I find accessible regions, I design the antisense oligos using good criteria as far as the oligonucleotide is concerned, like GC content, making sure it doesn't self-dimer and that sort of thing. And so it's hard, it is difficult at this point. And this is one of the reasons why there hasn't yet been a good algorithm to predict antisense design, or to predict a good antisense oligo, basically.
There's a certain amount of art to it.
There is, and it takes a while to design these things, but we are currently trying to train an SVM right now in order to design these more efficiently.
Cool. Another thing is that you earlier on were discussing what your lncRNA targets were, and you were mentioning the lengths of them. Is there any significance to the length of the target?
Well, there can be. So for messenger RNA, when you knock down, when you cleave it, it makes a non-functional protein. And so having retained fragments isn't as detrimental to your experiment. These long non-coding RNAs, however, a lot of them have structural significance in the cell as far as their function is concerned, and these structural components of the long non-coding RNA can bind proteins or can be potentially difficult for them to be degraded naturally after they're cleaved by RNases in the cell. And so the length this is one thing that we've kind of thought about, in that, let's say that for Cas7, you cleave it at the 3' end, but however you have a retained fragment at the 5' end, which is still functional. That might give you an erroneous phenotype as far as knocking down a long non-coding RNA goes. And so for the longer ones, I mean, some of these are up to 80 kb in length. You might have to target several sites simultaneously in order to break up those structures which might be functional if you're looking at a particular phenotype. Okay. However, saying that, again, we designed qPCR assays at both the 5' end and the 3' end, and for all of the targets studied here, we found they were completely degraded at the time that we took RNA. So while in this cases it didn't seem to matter so much, if you have the longer non-coding RNAs, it could potentially impact that.
Okay. Let's see here. This is an interesting question. Do you observe any correlation in the amount of knockdown that you get and the basal level of expression that you started with?
Sometimes. So if you remember NRON, which is our lowest expressing non-coding RNA, this is the one that we were unable to knock down. And the first time that I presented this, there was another scientist at Stanford which approached me and said that in their lab they find it sometimes difficult to knock down long non-coding RNAs which have low abundance. However, hot air was probably, all of these other ones were very, very highly expressed. Hot air was borderline, and so this had pretty low expression, but we were still able to knock it down quite consistently. So I'm not sure if that's, in general, I have heard that to be true. In my hands, we had one low abundant one that was not able to be knocked down, while we had another one which was able to be knocked down.
Okay. So I actually have a pretty, this is a basic science question. You covered a lot of modifications and stuff in here. Somebody was asking just what a LNA was.
Okay, so an LNA, it's a locked nucleic acid. It's basically it's a modification which has a methylene bridge between the 2' and the 4' carbon on the base. And more importantly, what it does is it increases binding affinity, and it also can have some sort of nucleus stability, not as well as some of the other modifications, but it's generally used to enhance binding affinity. And when you do that, it allows you to shorten the oligonucleotide, and so that's why these ones are about 15 to 16 mer in length. Shortening an oligonucleotide can help with cellular uptake, as well as it can minimize the cost of producing it. But basically, that's what LNAs are used to enhance the binding affinity of an oligo.
Okay. And then another one was, can you explain what a chemical match is when it's used as a negative control?
Sure. So for all of the DNA phosphorothioate antisense oligos, the same sequence, negative control sequence, was used. There were 2 that were used for the antisense oligos, and we compared the DNA phosphorothioate antisense oligos to a negative control that was all DNA phosphorothioate. We compared the 2'-methyl gapmers to a negative control, which was a 2'-methyl gapmer. And so basically we're comparing the exact same chemical modification with its negative control sequence. And the same is true for the siRNAs. The siRNA 21 mers was compared to a negative control, which was an siRNA 21 mer, and the dicer substrate was compared to a negative control dicer substrate.
Okay. Can you go to slide 22, Kim?
Sure.
This person's asking, by combining, they want to know if you mean targeting the exact same location, or would you be targeting different locations?
Nope, these are targeting different locations. So we selected one of the antisense sites that we already tested that I showed in the previous slides, and one of the dicer substrate sites. And I didn't select the most active antisense site because I wanted to see if you could further enhance activity by combination. But these are 2 different sites that are used, and they were co-transfected together using lipofectamine 2000.
Okay. And what is the time point for the knockdown?
24 hours.
Are they all 24 hours?
They are 24 hours. I also did them at 48 hours, and I got the same results.
Our friend Matt asked a question. He previously, and now somebody else is asking this question. Do you sometimes see an increase of the target with treatment of the antisense oligos? And can you explain that? I can't remember what slide that is on.
Sure. So here's an example right here where, and this is all kind of, it's dependent on your negative control that you're comparing it to, where sometimes you see an increase in activity above 100%, but we consider this to be all kind of in the noise level and not real. Like, we don't believe this to be an increase in activity upon treatment. More so, it's just how all the data is normalized, and once you get up into these levels, it's more noise. That's the same reason why when we look at let me go back to, when we look at these levels, for example, for NEAT1, and you see, oh, you get 20% knockdown, this is all kind of we consider in the noise, and we don't start to really trust the numbers until they're down in maybe the 60 to 90% knockdown range.
Okay. So this person's asking something. I'm not sure. Maybe this is something we're doing some research with, or know somebody who's doing research with. What do you think about doing gene knockout for long non-coding RNA using CRISPR? Do you know if anybody's doing that?
Yes, I think it.
That might just be something we have to we can reply to them, later on, see if we can find an answer for it. It's a little off topic.
It's difficult because you can still create these functional domains, like depending on where the CRISPR is designed, right? So for messenger RNA, it's going to disrupt a protein being made. But for the long non-coding RNA, you could still potentially transcribe short functional domains. And so from what I understand, using CRISPR to target long non-coding RNAs is a lot more complicated. And yeah, if you get their email address, I can send them a paper at a conference that I just went to of some of the tricks that they were using in order to use CRISPR to knock down a long non-coding RNA.
We do have some attendees that have commented that there are some genome-wide ones apparently that have been done. So yeah, we'll have to look into that. There's a dose question or two in here. So is there any need to try more than a 10 nanomolar dose for the antisense oligos?
If you're just looking at knockdown, it's nice to see a dose response, and that's what, like, if you plan on publishing, people will kind of expect to do a dose response. Usually for these antisense for the long non-coding RNAs, we use a 10, 3, and 1 nanomolar dose to show kind of that. I mean, it shows that you're getting expected results basically, and not something that could be caused by potentially toxicity or an off-target effect. And so, yeah, we do like to use at least a 3-point dose response.
So another person was saying that they had used significantly more. They'd used like 50 nanomolar and 100 nanomolar, and they didn't actually see any effect, and they want to know if they should maybe go back and try those lower doses.
If it was non-toxic and you don't see a result at 50 nanomolar, I doubt that you would see a result at a lower dose. So this is also cell line specific. I mean, different different cells have different levels of ability to transfect. HeLa cells are very easy to transfect, and so are the HGT116 and HUH7. You get very effective or efficient transfection. Some cell lines, if they're more difficult to transfect, you might have to use a higher dose because it's basically how much actually gets into the cell. And so that's a factor to keep in mind. You probably need a positive control in order to show that your oligo is taken up. If you remember from NRON, it could be a long non-coding RNA, which is just difficult to knock down, or you might need to try some additional sites. And so when I'm designing antisense oligos for customers, I recommend testing at least 5 different sites in order to get a good hit. And if within those 5, none of them work, which I don't actually think I've never received feedback of that happening, but if not, I would recommend doing 5 more. And then if that didn't work, it might just be a long non-coding RNA, which is refractory to being knocked down.
Sure. So rather than change the dose, basically they should go back and try some other sites. That's the
first verify if they had a positive control to show that knockdown was achievable by using their particular transfection reagent in their cell line. And then if they do show that, maybe redesign the antisense oligos. And they can contact our application scientists to
Sure.
help with design.
And somebody else asked, is there a preferred transfection reagent? Which again, I suppose would be somewhat cell type specific, but do you have one that you prefer to use?
It's absolutely cell line specific. And in cell lines that are easy to transfect, like these HeLa cells or HET116, we found that the antisense oligos work exceptionally well with lipofectamine 2000. The only drawback is that lipofectamine 2000 can sometimes cause toxicity. And so we also use RNAi max. Usually for the RNAi reagents, we like to use RNAi max from Life Technologies as well. But we found that lipofectamine 2000, we didn't see any difference in activity with the RNAi reagents compared to RNAi max. But it is absolutely cell line specific, and again, they can contact us with their particular cell line, and if it's one that we use in-house, we can give them a recommendation as to which transfection reagent to use.
Okay. So this person's asking something specific that might be of interest. They're targeting a long non-coding RNA that's localized on the positive strand. However, there's another long non-coding RNA that's on the negative strand in the same area. And does that mean that they can only use an antisense oligo to knock down that lncRNA?
To be safe, I would say yes. The chemical modifications here, like for example, 27 or 21 mers unmodified can load both strands, the passenger and the guide strand. The dicer substrates here are chemically modified to eliminate passenger strand loading. And so in all the studies that we have done, you don't get any activity from the passenger strand, only from the antisense strand. And from what I understand, the LNA modifications for the silencer select siRNAs serve the same purpose, to only for strand selectivity, so it only loads the guide strand. To be on the safe side, just in case, an antisense oligo would absolutely only target one of the strands.
Okay. This is an interesting question. So this person is wondering if we have any experience with circular RNAs, and then they have, is it putative microRNA or?
Sure.
Okay.
Sure. So that was one of them that was actually going to be originally in this study, but we thought that because it's a cytoplasmic, the one that's the miR7 sponge, because it's a cytoplasmic non-coding RNA, I was going to include it in our study here, but we thought that it might confound things a little bit too much, as you know, now you're adding not only just different localizations, but also linear versus circular, and the fact that it does bind miR7, does it bind a whole bunch of proteins. And so just for this original study, we did not test a circular non-coding RNA. However, that is one of my intentions, to go back after this study is complete and look more at the different kinds of long non-coding RNAs. So no, I have not knocked it down.
Okay. We have lots of questions here. It's taking me just a little bit to read through them. They're great questions. I'm very impressed with the good questions we've gotten. All right, so this person's question is, my understanding was that knockdown of nuclear transcripts with siRNA was difficult or impossible given the absence of risk in the nucleus.
Well, that was a general thought, but the Corey lab published a paper in 2014 in Cell Reports, and its RNAi factors are active and functional in the nuclei, and he showed very convincing evidence that with the exception of the loading factors for risk, all of the other risk components are in the nucleus, and he showed very convincing data that MALAT1 was able to be knocked down both in the cytoplasmic fractions as well as in the nuclear fractions, and also showed in situ hybridization data after knockdown with an siRNA that MALAT1 disappeared. And so we appreciated that going into the study, and that wasn't really the objective of the study, was to prove that it was in the nucleus or not in the nucleus. We just kind of wanted to give an overview as if you had to choose a method of knockdown, which one would you choose to have the higher level of success? And one of the things that I didn't mention about the cytoplasmic targets is that RNAi factors are found to be in P-bodies, but they're found to be functional at the rough endoplasmic reticulum, and that's where translation occurs. These two long non-coding RNAs, DANCR and OIP5AS1, are both associated with heavy polysomes, and so they are in the proximity of the RNAi factors in the cytoplasm. If these were, if the long non-coding RNA isn't around the heavy polysomes or around the rough endoplasmic reticulum, I'm not sure how effective RNAi would be in the cytoplasm either. And so that's one thing that we're going to also look at is do different localizations in the cytoplasm affect the ability to knock down with RNAi. But yeah, so look up that paper in Cell Reports in 2014, and the data is very convincing that RNAi will work in the nucleus. The question is does it work better in the cytoplasm versus the nucleus, or for long non-coding RNAs, does it matter?
That's pretty cool,
a good question. Yeah.
Yeah, cool. All right. All right, the next question, this is a very good specific question, is your qPCR assays to measure your knockdown, are they designed for the binding site of your knockdown, your ASO?
No, I understand the question. So no, they are not, because for two reasons. One is that, let me pull up one of these. We would have to design, potentially 33 different qPCR reactions. And so what we did instead was we designed one at the 3' end of the target and one at the 5' end of the target. And we ensured that we had consistent, concordant results with the two assays. So the two assays were multiplexed together for every single target for every single knockdown reagent. And we were convinced that because we got the same results with the 5' qPCR assay and the 3' qPCR assay, that it limited the retained fragments in the middle, or it at least eliminated any sort of erroneous amplification.
Okay.
For NRON, for this target here, I did go back and I designed 6 qPCR assays, which encompassed most of the binding sites to be sure that that we weren't knocking down NRON and just having all retained fragments because of the protein binding or because of a particular secondary structure of the target. And with all of the 6 different NRON qPCR assays, I did not have any knockdown.
Okay. So if it's difficult to knock down long non-coding RNAs with low expression levels, would it be better to knock out those lncRNAs? Is that possible?
Yes, it should be possible. I mean, it's going to be more time consuming, clearly, but I have not done any knockout experiments myself. And so that would be something to look into if you're unable to knock it down by these RNA knockdown techniques.
Okay. You may have addressed this. I'm not sure if you did. Do you see any difference in cellular context regarding the same locked nucleic acids?
Can you repeat the question?
They're asking, do you see the cell context difference with the LNA reagents?
Meaning
Yeah, I'm not entirely sure.
that that the LNAs worked better in one context versus the other.
Yeah.
I would say overall no. There didn't seem to be a striking example where the LNA worked a lot better in the cytoplasm versus the nucleus. They all performed about the same. And just keep in mind that the antisense oligos can target a cytoplasmic RNA before it even is exported out into the cytoplasm. So that's one of the advantages of antisense oligos, is that if they do function mostly in the nucleus, it can target RNAs before they're exported out. But no, I did not see a pattern between the LNA-modified oligos as to whether it outperformed one or the other depending on the cellular localization of it.
Okay. I just want to comment to people. I noticed that a few people had asked for citations that Kim has mentioned. We'll follow up with you by email and address your request specifically that way. Yeah, okay. So somebody is interested in the lncRNA fast response, that early response that you get, and for example, in like 30 minutes. Should we start the knockdown 24 hours ahead of time, even when the lncRNA is not expressed? So can they introduce their reagents before the event that's going to trigger their lncRNA expression?
Yes. Yes, they can. And in fact, that will ensure that the antisense oligo or RNAi reagent is loaded and ready to go for whenever are they turning it on? Are they inducing it?
They weren't specific. They just said they're
Yes,
interested
you can
in that
pre-treat.
early response.
Yeah, you can pre-treat. We see antisense activity basically will be consistent until for these armored antisense oligos, meaning they're not easily degraded by nucleases, we'll see activity until the cell divides enough that it dilutes out the antisense oligo. And the same thing is for a good RNAi reagent. For a dicer substrate, we'll see knockdown for up to 5 days. So yes, you can pre-treat your cells before you induce expression of your long non-coding RNA.
Okay, here's a design question for you. So given the fact that most lncRNAs have different splicing forms, what's the best region to design to the target? Would it be 5' or 3'? Does it matter?
Well, that's long non-coding RNA specific. You'd have to map out the different isoforms, and then we would hone in on the region that if you wanted to knock out the entire thing, the region that was consistent between all the different isoforms. And if you were looking to be isoform specific, we would try to find a region which was not retained in all the different isoforms, if that makes sense. So I wouldn't say 5' or 3' end is necessarily different. We just look at the entire sequence of all the different isoforms and try to design it intelligently that way.
Is there any data for the effectiveness of these methods in non-adherent cells in suspension, since they're difficult to transfect?
I have not tested them in suspension cells. Most all of our cell lines that we typically use here at IDT, they're adherent. And so, yeah, but it would be the same for an adherent cell line, which was difficult to transfect, that basically optimizing the transfection is the first step in these knockdown experiments, and you can do so by targeting a positive control. And we have positive controls available on our website, or you can choose your favorite one for being able to test transfection efficiency. But we do like to a positive control instead of just a dye-labeled control, because we have found that dye-labeled controls can be taken up into the cell, but not necessarily in a compartment that is necessary for your particular knockdown, or they can maybe stick to the outside of the cells and cause fluorescence that way, which makes you think that they're inside the cells. And so we actually like to use a positive control that targets a gene, and then we use that for as far as transfection efficiency standards go.
Okay.
I don't know if that answered.
It seems to mostly answer that. Yeah, I think so. We'll find out. We'll follow up with people after this and make sure that your questions got answered. And you can always contact that application support email address. I don't know if you want to put that up really quick, Kim, just so
sure.
can see that again. And then while you're going to that resource slide, I will ask the next question too. So you measure the time point at 24 hours. How long after that does the knockdown last? Does expression resume after some period of time?
It does. I have also tested it at 48 hours, and then for NRON, I also tested it at 72 hours. I was really determined to get NRON knockdown. And we do see it pretty consistent up for a couple of days. I have not tested, I've not done studies which went out past 72 hours with the antisense oligos for long non-coding RNAs at this point. So that would be something that you'd have to test. And it also depends on how rapidly your cells divide, because they're eventually going to dilute out that antisense oligos too. So for rapidly dividing cells, the effect would not be as long as for non-dividing cells.
And I think this is a very similar question. Transfection is transient, so how long will the siRNA work and inhibit signaling?
I've not done that for the long non-coding RNAs, but for a messenger RNA, for the dicer substrates, we have seen, for unmodified dicer substrates, we've seen effects lasting up to 5 days. And after 5 days, that's when RNA levels start to come back. And that was done in HeLa cells, so those are considered to be rapidly dividing cells.
Okay. Do you know anything about this, Kim? Could you comment on nuclear leashes on oligos and their design and benefits for knocking down nuclear non-coding RNAs?
No, I don't know. Nuclear leashes?
Yeah, I'm not sure. That's a new term to me as well. So that's something we can, again, follow up with, see if somebody.
Yeah, I'll have to look into that. No, I'm not familiar with that.
Okay, here's another question. Do you think shuttling of lncRNAs in the nucleus and cytoplasm is accounting for MALAT1 knockdown by RNAi?
Potentially, absolutely, because there is, at the recent conference that I went to, someone gave convincing data that there was about 10% of MALAT1 that was found in the cytoplasm. And if you look at my in situ hybridization results, if you don't mind me going back to those, I saw some MALAT1 that was also in the cytoplasm. You'll have to excuse this one because this is a dividing cell, and so all the RNA kind of pours into the cytoplasm when the cell is dividing. But if you look down here even, you can still see slight fluorescence here in the cytoplasm. And so it could be, however, again, if you look at that publication in Cell Reports, the RNAi factors are found in the nucleus. In that paper, they have a really nice image that shows, and this is ongoing right now, these are the experiments that I'm actually doing this week, that shows that you have an in situ signal in the nucleus, and then when you treat it with an siRNA, that the signal in the nucleus disappears. Whether or not it's being exported out and exported back in, and that's when it occurs, I'm not sure. I don't know the answer to that.
It's a very good question, for sure.
Yeah, it's a very good question, and it's a question that people are trying to figure out right now.
And I'm going to ask one more question, and I think this gets us through the very great majority of these, which is, have you tried targeting introns using antisense oligo?
I have not tried targeting introns using antisense oligos. Although it's been done more successfully, I was actually trying to find whether an RNAi has been able to target introns, because there's been a couple publications in 2002 and 2003 which show that they have not been able to target introns with RNAi. But as far as antisense goes, I have not, but there shouldn't be, I wouldn't think that there would be any complications with that.
Okay.
And you know what, I have designed antisense oligos for customers targeting introns, and they have worked successfully. So while I have not done those experiments, I have had customers tell me that they've had success with it.
So it's possible, and there should be some published data out there somewhere. Okay, so with that, this was a great set of questions today. I just want to thank everybody for attending and participating. Kim, thank you for the presentation and for taking everybody's questions. And yes, we have recorded today's presentation. People are asking if we'll be sending links to that. We'll send you a follow-up email to have a link to the presentation on our YouTube channel, and you'll also get a link to the slide deck itself, so you can go back and look for this application support email address or these associated articles and whatnot, whatever resources you want to find from here. For those of you that have requested that we send you a citation or something, we will follow up with you by email. And everybody will get some sort of follow-up to make sure that we answered your question. So yes, thanks again, everyone. This is one of a series of webinars that we'll be doing on RNAi and antisense oligo methods, as well as other molecular biology topics. We'll email you about those future webinars as they're scheduled. And you can find previous webinars and this webinar on our YouTube channel, which is www.youtube.com/IDTDNAbio. And our SlideShare site, which also has slide decks from previous webinars, is www.slideshare.net/IDTDNA. And like I said, we'll send you links to those for this presentation. All right, thanks again, everyone. That was great. Have a good day.